When we pick up a household cleaner or a prescription medication, we assume someone, somewhere, made sure it was safe. We expect a rigorous, reliable testing process before any of these chemicals reach the market. But this FDA presentation outlines the reality of the pre-1970s testing era. There were no published regulations for how laboratories conducted non-clinical studies, and no systematic evaluation to keep them honest. Government agencies like the FDA and the EPA had to rely entirely on the manufacturers. When a company wanted to sell a new pesticide or food additive, they submitted their own safety data, and regulators largely took them at their word. The entire public safety net rested on a foundation of blind trust. Without a way to verify the laboratory data, consumers were exposed to hidden systemic risks. In the mid-1970s, Industrial Biotest Laboratories, or IBT, was a dominant player in the testing industry. They were personally responsible for roughly 40% of all U.S. toxicological testing for major consumer and industrial products. In 1975, the FDA and EPA initiated an in-depth investigation targeting 40 different toxicology labs across the country. What they found at IBT was a culture of widespread fraud. Investigators discovered resurrected lab mice. Animals that had been officially recorded as dead in long-term studies somehow generated healthy data points days later. The fallout was massive. Due to fabricated data and untrained personnel, 75% of over 900 IBT studies had to be invalidated. Regulators were faced with a terrifying realization. Hundreds of chemicals were already being used in homes and on farms, approved based on data that had been lied about. Congress convened a series of intense hearings between 1975 and 1977. Lawmakers and regulators recognized a hard truth. Unstandardized, unverified scientific data is functionally meaningless. This timeline shows the exact publication of the resulting rule. In December 1978, the government finalized the Good Laboratory Practice Regulations, codified as 21 CFR Part 58, to secure the integrity of the testing process. A core pillar of GLP is accountability. Observations must be recorded in ink. Mistakes cannot be erased. They must be crossed out transparently, signed, and dated. GLP also mandates reconstructability. Detailed records must enable a third party to rebuild the entire experiment years later. Reaching a correct scientific conclusion was no longer enough. Researchers now had to prove exactly how they got there, leaving a permanent, transparent trail. GLP principles extend directly into the physical workspace. Facilities must be specifically designed to separate test systems and prevent cross-contamination, with restricted entry into animal rooms or sensitive areas. The equipment itself falls under heavy scrutiny. Instruments must be strictly maintained, regularly calibrated, and thoroughly documented to ensure precision. To enforce these rules internally, GLP requires a quality assurance unit. This is an independent watchdog group built into the organization, mandated to witness and audit the research as it happens. This presentation outlines the role of the external enforcement arm, the FDA's Office of Study Integrity and Surveillance. OSIS scientists verify data quality, assess compliance, and directly investigate complaints of falsification. By auditing the physical environment, the laboratory tools, and the people conducting experiments, GLP transformed vulnerable laboratories into secure, verifiable spaces. The demand for trusted data quickly expanded beyond U.S. borders. As international chemical trade grew, the global market required a unified standard to ensure safety without restricting commerce. This slide shows how the OECD developed global GLP principles in 1978, using the original U.S. rule as their foundation. This led to the Mutual Acceptance of Data, or MAD. Under this agreement, a compliance study conducted in one member country is accepted globally by all other participating regulatory bodies. This system eliminated the need for redundant trials. It removed a massive source of waste, cutting costs and lowering the number of animals used in testing worldwide. GLP created a universal scientific language. Nations can now share vital safety data openly without compromising their own domestic health standards. Today, those protocols act as an invisible armor for the public. Every pesticide, cosmetic, and pharmaceutical product on a store shelf is backed by this rigorous testing framework. The IBT scandal ended the practice of taking laboratory results on faith. Today, safety is defined by a permanent, verifiable trail of evidence.